Good morning. We have a lot to cover this morning, so we want to get started. First, I'm getting many emails regarding 131A, and they're wondering what to do with the last lecture because they can have typed up all of the other lectures, but the last lecture, what you do is just write in hand when you're finished. So you turn it in on Friday right after the lecture. Is that okay? I can see some of you still look puzzled. Is that clear? Fine. Good. Well, let's continue with the female reproductive system. And we worked our way down to the vulva, which is the external genitalia for the female. And we'll have the mons pubis, and if we put the mons pubis in here, it will develop. It represents a superficial fat pad over the pubic symphysis. The mons pubis is a superficial fat pad over the pubic symphysis, which you remember from our bone lectures. And then we'll have the labia majora, the large lips, labia majora, and these represent folds of skin. Folds of skin with their fat in the folds, muscle, glands, and connective tissue. And inside we'll have the clitoris, which represents two erectile bodies, two erectile bodies, but they create a single projection. And this erectile tissue, upon stimulation then, will increase the dimensions of the clitoris. So it increases dimensions with stimulation. What is the homologue in the male? The penis. Homologue in male. But she has no urethra in clitoris. Now coming from the clitoris then, we have the labia minora, labia minora. I forgot to put a hair on the majora. also has hair, because the labia minora has no hair, but these will be folds of skin with glands. The labia are to protect, then, both sets. Labia will protect the clitoris, the urethra, which is just posterior, we'll put a U, and the vagina. Protect the clitoris, urethra, and vagina. So that gives a very brief introduction to the external genitalia of the female. With that, another brief introduction to the breast. The breast will cover over the 2nd to 6th rib. It's over, we'll just put sits over 2nd to 6th rib. It is attached to the fascia of what muscle? Pectoralis major. Attached to fascia of pectoralis major. Now, it has connective tissue surrounding and connective tissue partitions forming lobes. So this would be, can't do it upside down. So lobe. And within the lobe will be secretory glands. From cells which are called alveoli. We've encountered them before. So we'd see the alveolar cells here forming into a duct system, and the duct converges at the nipple. These represent alveoli, and we could do them in color. And the ducts then coming to the nipple. The size of the breast and its shape is due not to the glandular material, but due to what? Fat. That gives the size. Quite interesting, isn't it? You can have a fairly flat breast and have lots of glandular material and be able to nurse your child. It's not due to the fat. All right, let's now then look at the male reproductive system. So the primary sexual organ for the male reproductive system is the testis. Primary sex organ equals the testis. That's singular, plural, testes. And these then are the gonads for the male, just to give the terminology, because it's used interchangeably. And the purpose of the testis then, the function, to form sperm and produce testosterone. So where are they? They're housed in the scrotum, which is a skin pouch. Housed in scrotum, which equals a skin pouch. And as we've said previously, that they prefer a cooler temperature to the body. What's your body temperature on the average? 98.6. Do you know your body temperature? No? Have you ever had it taken? Some have, some haven't. 98.6. That's sort of average body temp. And the testes prefer it 3 degrees cooler, 95.6 for testis. Has anybody found out why for us yet? Why they prefer it cold? What if it gets too cold? Have a safety valve. They have the cremasteric muscle to draw the testis up to the body. If too cold, like this morning, well, I put on my wool coat for the first time. I was really cold after swimming. We swim at 6 o'clock and it's so freezing. Anyhow, we're talking about the cremasteric muscle. If too cold, cremasteric muscle contracts and raises testis to body wall. So, now, let's begin with the structure. First, we have to get some stem cells. So roughly, they say that about 1,200 stem cells are formed in the embryo. destined for forming the testes. What will be forming these stem cells? The same structure that formed the ova. What was it? The yolk sac, right. Formed by the yolk sac. And they will then migrate to the site of the testis formation. There are names for all of these. I'm giving you the general principles here. And there, these stem cells will start the development of the sperm. And the sperm are going to develop in the so-called seminiferous tubules. We'll first show us the structure of sperm, testis. We'll again have CT capsule. And we'll divide into lobules. We have CT partitions again. And this will be a lobule. There'll be hundreds of lobules in the testis. And each will contain 2 or 3 seminiferous tubules. So we've got seminiferous tubules coming up. Seminiferous tubules. And their purpose then is to form sperm from the stem cells to start with. And forming sperm is referred to as spermatogenesis. Spermatogenesis equals spermatogenesis, formation of. And the sperm will be called what? Spermatozoa. So let's look then at a cross-section from one of our seminiferous tubules here. And we'll see the germinal epithelium around it coming up from the baseline. Germinal cells. They'll be changing their characteristics as they're forming the sperm. And eventually you'll have a sperm. Just different developmental stages have spermatocyte 1, spermatocyte 2, spermatogonia, and so forth. Each one has a name coming up. But I just wanted you to know the principle of developing from the baseline to form sperm. And also within the germinal epithelium, we have what are called the Sertoli cells. Sertoli cells. After Mr. Sertoli, so it's capitalized. And they extend from the base of the tubule all the way up. This will be our Sertoli cell. And they will be supplying nutrients and maintaining the hormonal level for these developing sperm. They form what's called the blood-testis barrier. We've had a blood-brain barrier. Now we have a blood-testis barrier. Because the internal milieu for the seminiferous tubule is different from the surrounding tissue. So it's important to have the Sertoli cell monitor such hormones, monitors concentration, of androgen, and estrogen. In the development of the sperm. So that gives you an idea of what's going on to develop our sperm. Let's be sure I've caught it all because I don't want to shortchange. Yes, also the Sertoli cells, then since these developing sperm will have sperm cells, will have cytoplasm. But by the time you get up to where their final stage as sperm only, the cytoplasm is shed, and the Sertoli cells then will phagocytize the cast-off cytoplasm. Sertoli cell phagocytize cast-off cytoplasm. So now we have a sperm. What are the basic structures in the sperm? Fairly clear-cut. We have a head, a body, and a tail. So this is our head. What's the matter? Does he look funny? All right. Head, body, tail. So, what is the head representing from the cell? It's the nucleus. And it will be carrying 23 chromosomes. 23 chromosomes. The body, what does it have? Mitochondria. It has spiraled mitochondria. The mitochondria then will provide the energy for the movement of the tail. Provides energy for movement of tail. And then we have the tail, which will propel, excuse me, propel the sperm. How fast do you think it propels it? Goes about 3 millimeters in an hour. As we say, not too swift. Now, in addition, the head has a component. I don't have any red chalk. I could make it green for Christmas. This is called the acrosome. Acrosome. Why do we need an acrosome? It contains the enzyme to allow the sperm to penetrate the ovum. So it contains enzyme for sperm to penetrate the ovum. So, remarkable little structure, isn't it? Now, what is the hormonal regulation of the testis? We've had it, but we'll put it in again. Hormonal regulation of testis, or testes if you like. So we have the seminiferous tubules that constitute our testis. So we have lots of sections of these. And we have interstitial cells. Interstitial between the seminiferous tubules out here in the connective tissue bed. So forth. These are interstitial cells. So, what hormone will be acting on the seminiferous tubules from the anterior pituitary? FSH. And the tubules then will produce sperm and Sertoli cells. What do we have acting on the interstitial cells? LH. And what will the interstitial cells then be producing? Testosterone. So you have both the main functions of the testes, testosterone. Testosterone becomes active, very active, at the time of puberty to make the masculine changes in the body. Just a few of those. Well, first, testosterone can be acting at the larynx. You've now learned the thyroid cartilage increases in size. Because the vocal cords are within it, and therefore changes the voice. Can be acting on the prostate gland. To become active with its secretions, which we'll see in a moment. And the seminal vesicles will be activated. All of these are going to be forming the semen, but those are examples other than the major body changes. All right. So with this, let's now look at our duct system. We'll take the sperm through the duct series. So the ducts, first one will be the epididymis. Epididymis. What in the world does epididymis mean? What's epi? Upon. What's didymis? Testis. So if we have our testis here, and we're producing all these sperm coming up, they'll come into, there are lots of names for all the ducts here, which you'll get in your advanced courses, but we're going to just take them to condense into the epididymis. Which will be sitting wound here on top of the testis. How long is your epididymis? It's 20 feet. Rather amazing. Who wants to help me? Will you help me? Come on. If I throw it out to you, can you wind it up and throw it back? It's easier if we do it right up here, because then one can see. Look at you. You will do it. Sure you will. One at each end. But I just think it gives you some visual perspective of this epididymis, which is wound up on this 1.5-inch testis. Isn't that amazing? Why do we have an epididymis? To store the sperm and allow them to mature. But isn't that amazing? All of that. I mean, what other structure did we have that was roughly this long? Small intestine. Right, and here we are, this little epididymis down on that little testis. Amazing. Thank you so much. Yes, they deserve a hand. They volunteered. Well, I appreciate it because I think it illustrates one good sight will last a lifetime, right? You'll think differently about your epididymi. All right, so they're being stored here. And they're being nourished. And the next tube that they're going to go into will be the vas deferens. You'll also see ductus deferens. Vas is vessel, ductus, duct, ductus. Same thing. And this tube will only be about 18 inches long. This was 20 feet. This is 18 inches. And it's a strong muscular tube. That will contract when one wants to ejaculate and help move the sperm along. Contracts with ejaculation. So now we're ready to come closer to our goal, which is the male urethra, to get these in position to leave the body. So let's look at the urinary bladder. And the urethra. And the, which gland are we putting in here? Prostate gland, good. Now, on the posterior surface of the urinary bladder are the seminal vesicles. And they sit like this. These represent seminal vesicles. And here comes our vas deferens, which will join with the duct from the seminal vesicles. Duct from seminal vesicles, and will form the ejaculatory duct, which will penetrate the prostate and bring the contents into the urethra. So in blue, we have the ejaculatory duct. And obviously you'll have the same on the opposite side. Vas deferens coming in, joining seminal vesicles and forming an ejaculatory duct. So now you can understand the relationship of the internal sphincter to the entrance of the sperm into the urethra, because our internal sphincter was up here between the prostate and the bladder. So when sperm are coming in here, this sphincter closes off and won't allow urine to go in. So this is the internal sphincter. All right, so that gets our duct system back into the ejaculatory duct. To make it complete, enters the prostatic urethra, just to give it more definition. We had given the various components of the urethra. All right, so that's our two of our duct systems. We've mentioned two of the glands, the prostate and the seminal vesicles. Let's see what happens with the secretions from the seminal vesicles or glands. Seminal vesicles. They'll account for about 60% of the semen. 60% of the semen. And they contain in their secretion a sugar, the fructose sugar. Fructose. To nourish the sperm now, they've left the epididymis. They're out on their own. Then we have the prostate gland. And it has a wide range for its contribution, some 15 to 30% of semen. And it will have an acid secretion. And the purpose of the acid secretion is to activate the sperm. So it's really on its own. Acid secretion, activate sperm. So it doesn't become active till it gets into the final duct system here. Then we have the, how big is a prostate? Prostate is walnut size. How big is a testis? Literature tells us 1.5 testis. 1.5 inches long by 1 inch wide. But there's tremendous variation in size. Much variation in size. So we're just putting this down at walnut size. Most people know a walnut. Thank you. And what is it secreting then? It's got its acid secretion. It's activating sperm. Our next gland will be the bulbourethral glands. Bulbourethral glands. And they are pea size, little fellows. Where are they found? They're in the urogenital diaphragm. If you recall where our urogenital diaphragm was, it was inferior to the prostate. We can just put it in. We had the urogenital diaphragm, which was skeletal muscle, your external sphincter. This was urogenital diaphragm. And they will have these little pea-shaped bulbourethral glands. Their secretion will neutralize the acid in the urethra. Neutralizes acid in urethra. So you can see what the contents of semen would be. They say single ejaculation, about 2.5 cc's to 6 cc's of semen in ejaculation. 2.5 cc's to 6 cc's of semen. But you now know what semen consists of. It consists of sperm, consists of the glandular secretions from these glands that we've been developing just now. So let's review if we can, please. All right, you can tell that this is the germinal epithelium, the outer line of the seminiferous tubule, and we have the different stages of development. Here's some sperm that have their heads in a Sertoli cell. Sertoli cell would be coming here, and the sperm then are going to be sloughed off into the lumen and carried through the duct system. In the next one, and what do you see here? Here they have red hats. What are they? Acrosomes, right? I didn't stain this one. It's the way it came. But you can see the development stages during spermatogenesis to eventually get a sperm. In the next one, and this is a cross-section through the epididymis, where you'll have this tube all coiled up, and it will have cilia on the epithelium, providing the nutrient to nourish the sperm. In the next one, and this is in the vas deferens, where we're seeing cilia here. In the next one, and this is vas deferens in a lower magnitude, but you can see again the amount of smooth muscle surrounding it. You don't see any other structure that has that much smooth muscle around a tube. In the next one, and then this is the penis. We didn't quite get there. We'll finish it next time, where we have the corpora cavernosa penis, these two cavernous bodies that are going to engorge with blood upon erection. But we also have the corpora cavernosa urethra, which will engorge with blood to a degree in the penis. Cross-section of a human penis. In the next one. And then this is the epithelium as we leave the duct system. In the next one, no, we'll skip. Let's stop because we've gone beyond. So we'll try to finish up early next time so we can have review. And re-embed and re-embed and repeat for you so you remember your anatomy. All right? Enjoy your afternoon.